Welcome to the Scat Track Field Collection Guide. Today, Ken and his trusted companion Helga will walk you through the steps for collecting wild canine scat samples for genetic and dietary analysis. Before heading out, make sure you have all the necessary equipment and materials in your Scat Track kit ready. Nitrile gloves, a surgical mask, forceps, labeled scat tubes, disposable sticks for sampling, and data card to record important information. And bring along a GPS device or phone app to mark sample locations. You can also download the SitSci app and follow the datasheet entry via the app, but fill out the hard copy as well. When you're in the field or walking down a trail, keep an eye out for wild canine scat. You'll often find it along trails, paths, or near areas with high wildlife movement and activity. Once you've spotted a sample, do your best to confirm it is from a wild canine by looking at its size, shape, and contents. For example, wild animals will have hair, bones, and other natural prey items in their scat, unlike a domestic dog that eats kibble. It will also often have a twisted end, unlike the scat of wild cats, which are more blunt. First, fill out your data card and SitSci app if you're using it. Be sure to include the tube identification, location coordinates, scat appearance, and any additional notes that might be relevant. Double-check that all information is accurately recorded, especially the tube identification that tracks the sample and sample location so we know where the scat came from. Before collecting the sample, put on a clean pair of gloves and mask. So that none of the samples are contaminated, Ken will make sure Helga steers clear of any scat he discovers, and he'll be careful not to come in contact with his pup during the collection process. Use the forceps to collect small pieces of scat from 2 to 3 different locations on the sample. Aim to gather scat that is roughly the size of a large pea. And try to avoid including bones, hair, or feathers, as they can interfere with our analysis. Instead, select fecal material that appears to be moist or less desiccated. Carefully immerse the pieces of scat into the buffer tube, making sure they are completely covered. This will help preserve the DNA. Remember, more is not necessarily better, so avoid overfilling the tube. Wipe the forceps in the grass to clean them, then place them, along with the tube and data card, in the small bag provided to mail back to Dr. Kristen Bresky. Keep the filled tubes out of direct sunlight and avoid high temperatures. The samples are stable at room temperature, so you don't need to refrigerate or freeze them for storage or shipping. Put any garbage in the larger bag, remove your gloves and mask, and use the provided hand wipes to clean your hands. To prevent double sampling in regions that are regularly visited, remove the remnants of the scat sample from the area once you've finished collecting. You can either kick it off the trail or mark it in a way that indicates it's already been sampled. And that's it. Following these steps will help us ensure high-quality samples for our Scat Track research. And remember, whether you're in the woods of Maine or Michigan, or the salt marshes of the Texas or Louisiana Gulf Coast, these methods are exactly the same. Thank you for your participation, and happy sampling.